Amino acids are the monomers for proteins, meaning that they're a constituent building block for polyamides. Amino acids are comprised of a central carbon atom, or the alpha carbon, with 4 bonded functional groups: a carboxylic acid group, a hydrogen group, a amine group, and a variable R group, which determines the type of amino acid that the monomer is. The one exception to this rule is proline, which is a secondary amino acid group instead. Additionally, amino acids are enantiomers, meaning that they demonstrate chirality. Chirality is a property of molecules in which they have mirror images of themselves that cannot be transposed onto each other. You can picture this as your left and right hand. Your left hand and right hand have the same general structure, but your left hand cannot be flipped to appear as your right hand. They are organised differently. The requirements for chirality are a central carbon atom with 4 atoms or functional groups attached, which must all be different. All amino acids except glycine have this property, and they each have 2 enantiomers, the lateral-rotation or left, and dextro-rotation or right. There are 20 amino acids, which are categorized into 4 categories. Hydrophobic aliphatics. These are repelled by water and contain no aromatic groups, and include glycine, alanine, valine, lysosine, isolysosine, methionine, proline, and cytosine. The hydrophatic aromatics. These are repelled by water and contain a benzene ring, and include tryptophan, tyrosine, and phenylamine. Polar charged. These are amino acids that have a hydrophilic R group and are charged at neutral pH, and include arginine, lysine, histidine, aspartic acid, and glutamic acid. The polar uncharged. These are amino acids with a hydrophilic R group and are uncharged at neutral pH, and include serine, threonine, glutamine, and asparagine. Proteins are polymers, consisting of a peptide bond, also known as an amide bond. Peptide bonds are created by condensation reactions, in which a hydrogen from an amino group of one amino acid and the hydroxyl group of the carboxylic acid of another amino acid is removed, creating a bond between the amino acids, forming a dipeptide and the byproduct of water. The bonds formed between amino acids can have a range of rotational or torsion angles. The torsion angle between the chiral carbon and nitrogen is called phi, whilst the torsion angle between the chiral carbon and the carbon in carboxylic acid is called psi. The physical possible values for phi and psi can be represented using a Ramachandran plot, which plots psi against phi, with shaded regions showing possible configurations. The covalent bonding between the individual amino acids created by condensation reactions comprise the primary structure of a protein. The primary structure of a protein influences the structure of the whole protein. The secondary structure of a protein is the folding of the peptide sequence into a regular shape. There are 2 most common folding shapes of a protein. The first is the alpha helix, an arrangement in which the oxygen in the carbonyl group of a carboxylic acid becomes hydrogen bonded to the hydrogen of an amino acid which is around 4 amino acids away, covering a distance of 0.54 nanometers. The other is a beta-plated sheet, which forms hydrogen bonds with other adjacent polypeptides. The adjacent polypeptide may be parallel or antiparallel with the other peptide depending on which direction the other polypeptide faces. The tertiary structure of a protein is the additional folding of the peptide structures from bonding that is not caused by the amide or hydrogen bonds, which may be disulfur bonds, interactions with hydrophobic or hydrophilic groups, and ionic bonds. Additionally, some proteins may have a quaternary structure involving interactions with different peptide chains. Electrophoresis is the separation of proteins by an electrical field through a polyacromide or agarose gel. Proteins are placed in wells and move through the gel in the direction of the positive anode, as they are negative. However, larger molecules experience a retardation force due to the mass caused by resistance against electrophoresis gel, and so travel less distance compared to smaller protein fragments. Electrophoresis may additionally be SDS electrophoresis, in which the samples are treated with a reducing agent and detergent sodium dodecyl sulfate, giving them an equally negative electrical charge and therefore only separating the proteins by mass. The aim of protein purification is to create a sample which contains only one specific protein, which can then be studied. The starting sample is typically taken from tissue samples, but may also be samples of bacteria which have been engineered by recombinant DNA techniques to produce more of a certain protein. The sample is then homogenized, or osmotic lysis may be used, in which the sample is placed in a hypertonic solution, causing the cells to burst. The sample is then centrifuged to remove cellular organelles. The proteins in the sample may now be isolated using many techniques. A common separation method is using ammonium sulfate, causing less soluble proteins to become precipitated. For example, in blood samples, a molar concentration of 0.8 causes the clotting protein flibberlogen to precipitate, and increasing this to 2.4 causes albumin to precipitate. Another method is using dialysis, in which small pores in a semi-permeable membrane is used to separate proteins by size.